Okay.
What kind of bird is that?
Duck.
Duck. So
Fancy duck.
fish-eating duck. We did see the harrier flew over us and looked at us if we were tantalizing.
Were you?
No, no, no.
Oh,
So Diane, how are you doing?
I am one day out of my surgery,
Oh my gosh.
so I'm home because it's
Yeah,
same day
that's great.
surgery. Yeah,
Good.
and I'm a little dingy, so James is gonna run this for me.
Okay.
But I'll be here because I'm kind of interested in iron and lysosomes.
Thanks for being here, Diane. I appreciate it.
Oh, yeah, I learned so much from you, Adam. I'm really happy to be here.
I was just thinking how much I miss all of you guys. Well, especially like you're still here, but Jerry and
I know,
the other day.
I know. That's true.
When does John is John departed already?
2
No.
weeks.
2 weeks.
I don't know if he's made that a public announcement everywhere. I know he's made it at the university.
It's not. Yeah.
Sorry.
Yeah.
Uh-oh.
So, no, it's okay. It's just,
All right. Should we start?
yeah, do it.
All right. So my name is James Cox. It gives me great pleasure to introduce my friend, colleague, and sometimes ski partner, when he can keep up, Adam Hughes. Adam, while originally from Kansas City area, Adam did his undergraduate work in Indiana University, Pennsylvania, majoring in biochemistry. And I think you were the first class out of biochemistry from there, right, Adam?
I think so. Yeah.
Yeah. From there, he obtained a PhD in biochemistry at Johns Hopkins, studying with Peter Espenshade, and had several really high impact papers, including publications in Cell and Cell Metabolism. He then got smart and moved all the way across the country to the West Coast, which is the best coast, the Fred Hutch, where he continued his studies with Dan Gottschling, really having a nice paper in Nature describing how early age increase in vacuolar pH limits mitochondrial function and lifespan. And this paper really set off his current research trajectory, which I think Adam will describe to us shortly. Adam has been quite successful early on in his career, winning a number of awards, including a Serial Scholar Award, the Glenn Award for Research in Biological Mechanisms of Aging, and my favorite, which I think you're the first one to have been awarded this, the Dana Carroll Endowed Associate Professorship in Biochemistry Award this year. Adam's well funded with the Mira and R1 awards, and with that, take it away.
Excellent. Thanks, James. And no, I can't keep up with you when we ski. Let's just be honest how it really goes. Can everyone see the other slide? Is the slide set up okay now? Just want to make sure.
Yeah.
I think we're seeing your presenter view.
Yeah.
You're seeing my presenter view?
Yeah.
Yes.
Okay. Better?
Yes.
Now you see the real one, not my notes? Okay, great. Thank you. Well, I'm excited to be here with you all on Zoom today, virtually. Of course, we've all done a lot of Zoom talks over the last couple of years, and I know that we're all fatigued. So hopefully we can make this fun today. So I would say I have the chat up. It gets a little clunky sometimes to try to monitor all of it, but please interject and interrupt and put things in chat, and I'll try to answer as much as I can, because I would love for this to be more of a discussion. And if it goes off course, so be it. And that would be great and fun. Because I know quite a few of you have seen some of this work before. A lot of it, some of the small details. So what I'm going to try to really talk about today is some of the broad strokes and some of the new, a little bit of the old and a little bit of the new directions where we're going, and just kind of thinking of bigger ideas. So I would love to hear some of your comments. So get this working. Okay. So our lab has many interests, but one of the interests is trying to understand and how our work related to metals and iron and the Center for Iron and Heme Disorders came up. Is we're interested in understanding how organelles within cells are functionally interconnected with each other, and how changes in these interconnections, oftentimes metabolic in nature, as I'll talk about today, serve as a driving point for aging and age-related disorders such as neurodegenerative disease, diabetes, cancer, you name it. So the two organelles that I'm going to focus a lot on today, the one that was in the title and the one that wasn't. So mitochondria and lysosomes. And I'll just say it up front, all the work I'm going to show you today, the predominant amount of work in my lab uses the model organism, budding yeast Saccharomyces cerevisiae. So in yeast, lysosomes are known as vacuoles. And there's some similarities and some differences, but by and large, I may use the words interchangeably today, but I'll highlight specifically when certain things that we're looking at are also applicable to mammalian cells. So let's first talk about mitochondria. Most of in this audience are very familiar with these double membrane organelles that are central to cellular metabolism. They play obviously many roles in the cell. Particularly relevant today is the production of iron sulfur clusters, as well as heme. They're also heavily involved in amino acid biosynthesis, lipids, apoptosis, obviously ATP production. The other organelle that we're going to talk about is the vacuole in yeast, so the lysosome and mammalian cells. So this organelle is involved also in a number of different cellular processes, right? It's canonically known as the cell's garbage can. That was how we knew of it for a long, long time. But in yeast especially, there was a lot of older work and really nice work suggesting that the vacuole plays many more roles in the cells. And so as I'll talk about today, in addition to its role in protein turnover, it plays a role in pH control, nutrient storage, which will become very important for today's talk. This could be amino acids, metals, ions, as well as nutrient signaling. So mTOR, other nutrient sensing pathways in both yeast and mammalian cells are located and interact with the vacuole or lysosome membrane where they often play a role in sensing metabolites. So most of the functions of the vacuole, I'll talk a lot about this today, are driven by a protein complex called the vacuolar ATPase. This is evolutionarily conserved and it's shown here. It pumps protons into the lumen of this organelle to acidify it. But the other major thing that this is used for is it's a proton gradient that's used for the movement of ions and metabolites across this membrane. This is going to be very critical to all the work that I'm talking about today. So our interest in understanding the interconnected relationship between these organelles stems back from work that I did as a postdoc with Dan Gottschling at the Hutch before I came here to Utah in 2014. And it had been known actually in the literature for over 30 years that these two organelles are functionally connected in cells. And the way that the connectedness, I would say, is an interconnected relationship where there's crosstalk back and forth. Our lab tends to study things in a unidirectional manner, just because I think that's how we came onto it and we haven't explored the other side. And what I mean by that is it's well known across organisms, and this is true from yeast to mammalian cells, and there's an abundance of literature, and I'll show you some examples in a moment, that the pH of the vacuole. So the bottom line is you need to maintain an acidic vacuole to have normal and healthy mitochondrial respiration. So if you lose vacuole acidification, as I'll show you, this can happen due to aging, due to mutations in subunits of the vacuolar ATPase, or other components, or oftentimes what we use a lot is chemical inhibitors or genetic inhibitors. So if this pH increases, what you'll see is that this, in many contexts, leads to an inability of mitochondria to function. It leads to a respiratory deficient phenotype. And so this was an observation, again, that had been in the literature much before my time, really probably before I was even born. And there was a number of different studies showing that alterations in the function of the vacuole would lead to rapid mitochondrial dysfunction. And as I mentioned today, two of the tools that we use and the way we got onto this was actually using a yeast aging system. So in Dan's lab, I was focused on using yeast as a replicative aging model. And we found that during the aging process, one of the things that happens is there's impairment of the vacuolar ATPase and acidification of the vacuole. And we found that drives a number of mitochondrial problems in cells and leads to mitochondrial dysfunction. So I'm going to show you a few different ways that we can actually look at this and see some of these phenotypes, and they're pretty striking. And a lot of these phenotypes, again, were shown before our lab, but I'm just showing you some of the ways that we've done it here. So this is a really good example. So this is a yeast growth assay here where we're spotting serial dilutions of yeast strain. This is a wild type strain here. And these are two different mutations in the vacuolar ATPase. And so what's really cool in yeast that allows us to study the VATPase and vacuole is that mutations and deletions of the vacuolar ATPase, at least in the strain background that we work in, which is BY4741 for your yeast aficionados, it's non-essential. I'm actually not showing that here. So if you grow them on glucose medium, the cells are sick, but they can survive, and we'll come to that in a minute. However, one of the most striking phenotypes that was observed in some of the initial screens for looking for mitochondrial respiration deficient cells is that VMA mutants, unlike a wild type strain, cannot grow on carbon sources like glycerol or ethanol, anything that requires respiration. So they have a pretty severe respiratory growth defect. This is something, so that was a genetic mutation that ablates VATPase function for a long time and shows that mitochondria function is impaired. There's other ways to do this too. One of the ways we use quite often is acute treatments, and we do a lot of imaging in my lab. So this is a time-lapse imaging movie of yeast mitochondria marked with a mitochondrial protein TOM70 that we use quite a bit. And I'm just going to play this here, and what you can hope to appreciate from it, this is just a vehicle DMSO treated cell. These mitochondria are dynamic. They undergo fission and fusion, but they have a largely tubular reticular nature that we've come to grow and love over the years. However, what's really cool is if you treat these cells with kincanamycin A, which is a specific inhibitor of the vacuolar ATPase over the same time course, the movie's a little bit glitchy today, but what you can see is that these mitochondria over time begin to fragment, and they ultimately end up in a more fragmented morphology. And what I'm not showing you here is this also correlates with the decline in the mitochondrial membrane potential. So the bottom line with this is loss of VATPase function leads to very acute and rapid mitochondrial impairment. And we went on to show, this is still back from when I was a postdoc, that this plays a major role in regulating mitochondrial health during the aging process. So in yeast, again, one of the ways in which you can study aging is by monitoring the number of times an individual yeast cell divides to produce a daughter. It leaves these bud scars left over on the surface. I won't talk too much about this today, but our lab does do quite a bit in this space. And what we observed is that if you look in old yeast mitochondria, the mitochondria are highly, unlike a young tubular one, the mitochondria are highly fragmented. And it turns out that what we showed at the time is that this is actually due to age-dependent loss of vacuolar acidification. It actually acts upstream of the changes in the mitochondria. And what's pretty cool is we can overexpress subunits of the vacuolar ATPase in old cells, and it actually prevents the loss of vacuolar acidification, and it prevents mitochondrial dysfunction, and furthermore, ultimately extends lifespan. And so this was when we were doing this work quite a few years ago now. The way we got into this was by doing genetic screens to look for things that could restore mitochondrial health in old cells, and that got us onto the VATPase. And then as I started to dig into it, I realized that we were really rediscovering in old cells here a phenomenon again that had been known for a very long time. And that is, again, that you need a functioning vacuolar lysosome to maintain mitochondrial health. And not only was this phenomenon known, it was actually well appreciated in multiple systems. And actually, it's been highly implicated in a number of age-related disorders. And so obviously, mitochondrial dysfunction and lysosome dysfunction in mammals are tightly linked to the aging process, but they're also correlated and show up all the time with a number of diseases like cancer, diabetes, Parkinson's disease. So you can find a million reviews that say, mitochondrial dysfunction, lysosome dysfunction are drivers of these disorders. And so I think the fact that this connection was there, we became very interested in. What we were really seeing in our own system was, okay, yeast is actually a pretty powerful system in which we can actually see this functional relationship taking place during an aging system and in an aging environment. And perhaps this actually has something to do with all these diseases that are co-linked to changes in the function of the vacuole and the function of the lysosome. So sort of because of those relationships, we became, in its applicability to disease, we became very interested in this question. And this has been a major focus of the research over the last several years. And this is what is going to dive into metal metabolism today, and actually quite a bit into amino acid metabolism as well. And that is, what is this relationship? So why do you need a healthy vacuole to maintain respiration? And so what I'm going to talk about today, just sort of a quick overview. So I'm going to tell two short little stories. The first one is published, and we published it, and this is work done by Casey Hughes, as well as TK Cody in our lab. And a lot of you in this group have heard this story extensively, and I'm just going to spend a little bit of time on it, just getting this up to the same page. And that is our findings that exploring this, that vacuoles and mitochondria are functionally linked through a really interesting mechanism that seems to involve an interplay between amino acid and iron homeostasis that I'll describe in a second. And then after going through some of this old data, I'm going to jump into some of the latest things we've been doing. And again, TK has presented from my lab, a graduate student has presented on this quite a bit to this group. And that is we're using genetic screens to identify cells that help, pathways that help cells adapt to changing vacuole function. So in other words, if you lose vacuole function, as I mentioned before, the cell can survive. But we're very interested in understanding what pathways are required to actually enable survival. And to get an idea as to how cells adapt to these changes in amino acid and iron homeostasis that are affecting mitochondrial function. And so in this part of it, I'm going to talk just a little bit about some of our recent work. I'm not going to get into all the details from TK Cody, a grad student who's near graduation, almost finished in our lab, as well as a new student, relatively new student, Kevin Choi, who joined the lab a couple years ago and has also picked up on this project. Okay. So when exploring these things, again, coming back to this question, how are these two organelles functionally linked? And this is all published data. And so I'm just going to kind of run through it using models and a few pieces of data here and there, but please stop me if something's not clear, more just to summarize. So coming back, when we started thinking about this, there's several different ways that we approach this, but in thinking about what the vacuole does, right? It does a lot of things. And I think we are first obviously drawn to its role in protein turnover here, which is actually X'd out. You know, the idea, and there's certainly a lot of evidence for this, that lysosomes in mammals and yeast, they're very heavily involved in autophagy and also degradation of proteins through other pathways. And I think the idea was that, okay, if you don't have a functioning vacuole, you know, you're not doing normal protein homeostasis, perhaps that backs up and impairs the function of the mitochondria, blocks things like mitophagy. And that is certainly the case and has a huge role in cells, especially more so in mammalian cells, I think. But one of the interesting things that we were able to do in yeast is we could eliminate protein turnover by knocking out proteases in the vacuole. And in these systems, there's still a proton gradient. And what we discovered from those studies was that if we got rid of the ability to degrade proteins, mitochondria were just fine. So the mitochondrial respiratory phenotype was actually linked to some function of the proton gradient, really, but beyond acidification. And so as I'm going to talk about today, what we really, through a process of elimination, really started to focus on the vacuole's role in nutrient storage and how important this was for preserving mitochondrial respiration. And so I'm going to take a little aside. I promise, I know this is a metals talk, and I will actually get to metals, but we have to get into the amino acid side of things first. And so what's really known, or really well known about vacuoles, and again, that we kind of re-got into, was that they play a big role in spatially organizing and storing amino acids within cells. So think about amino acids. This is really the predominant amount of work in my lab is focused on amino acid metabolism and amino acid homeostasis. And amino acids come into cells. This is a yeast cell, particularly same concept in mammalian cells. They come in through transporters, right? And then they need to be utilized. Different things can happen with them. And so the idea is that when they come in, they can be used for translation, they can be metabolized, but it turns out there has to be a balance. So I think in a lot of the ways, what we started to learn is how we think about glucose and lipids and how too much is bad. The same is true for amino acids, and we get into this more and more. And part of the role of what we think the vacuole does is it actually uses this proton gradient by the VATPase coupled to a series of transporters. And it actually moves amino acids, and this is very well known, into the vacuole, and quite a few of them are stored there. So if the cell is really well fed, the vacuole will have high levels of amino acids. If you're starved, this will work in the opposite direction, and amino acids will be spit out in order to be reused for translation in the case of nutrient scarcity. Through a number of different work in two different studies from our lab, what we ended up demonstrating and showing was evidence in our working hypothesis is that when you lose the proton gradient in the vacuole, one of the major effects this has on cells, if you think about this, amino acids are coming in, they're rapidly moving in here and exchanging. When you lose the proton gradient, these transporters shown in green here can no longer function. And so one of the things that happens, and this has been somewhat difficult to measure just because of the spatial compartmentation of it, but I'll show you a few different ways we've attempted this, is we think that there's at least a transient accumulation of amino acids, right? They come in, they suddenly can't get to where they normally go, and they accumulate in the cytoplasm, we think. And a few pieces of evidence for that. One of them is an RNA transcriptional response that we sort of think of as an amino acid stress response or an amino acid toxicity response. And what's pretty remarkable is within just a couple hours after you treat cells, a question or just a no, with VA2Pase inhibitors. One of the cell basically perceives that it is overloaded on amino acids. And so one of the things that it does is it represses, in blue here, most biosynthesis pathways for amino acid production. Not shown here, what it also does is it upregulates the enzymes that are involved in breaking down amino acids, a lot of transaminase enzymes that convert these amino acids, non-preferred amino acids, into glutamate. And we actually see that happening quite a bit here. So this is whole cell metabolite analysis that we've done. We've done a lot of work with the metabolomics core and James Cox. It's been a really fantastic collaboration over the years. And all I want you to appreciate from this is, so what cells tend to do, and this isn't too particularly important for the talk, but it just gives you an idea that cells are really struggling with amino acids. And what they like to do is they actually will take non-preferred amino acids and turn them into glutamate. Seems to be the predominant thing that's happening. So in a vehicle treated cell, here's sort of steady state levels of amino acids. If we treat them with concatamycin A, these are all normalized to 1. What we see is that over time, this is after 3 to 4 hours, the levels of most amino acids actually drop. So the idea is that they're in the cytoplasm, but what the cell is doing is actually changing these amino acids into glutamate and ultimately glutamine. And so we think there's a shift in what the cell is actually wanting to do here. And through a lot of work that I don't have time to show you today, we think the reason is that most of these amino acids are actually toxic for the cells for different reasons. And a large part of our lab is trying to understand mechanisms by which these amino acids become toxic. And as I'll focus on today, one of the amino acids becomes particularly toxic for the cell, and that's cysteine, and it does so by impairing iron homeostasis. But what's becoming cool and is sort of a theme is that cells really need to handle where they have amino acids and when and how many they have. And if this changes or there's a disbalance, they undergo a large number of adaptations in order to restore amino acid homeostasis. And I think part of the reason is that these amino acids have interesting properties that start interfering with other cellular processes. And we're going to dive a lot more into this today, and especially in the context of iron. Okay, so this is just showing this in model view, and a few other things we can do. And one of the cool things, so the idea is that during aging, what's happening? Question? No, okay.
So Adam, I'll ask a question. So on that last slide, you showed this major drop in amino acids, especially like lysine. Can you show that there's a growth defect by adding exogenous amino acids? They may not affect mitochondria, but is there a growth defect with an elevation in extracellular amino acids?
Yeah, so this has been really tricky, and I'll come to this at the end, Dennis. So let me go here because it'll be easier to explain. So this system, right, the bottom line is what we know now is there are multiple systems in the cells, and we've kind of jumped in on this from other labs that control amino acid homeostasis. So you can put them in vacuoles. Translation, which isn't shown here, is obviously a major consumer of amino acids. There's another thing in our lab studies called the mitochondrial drive compartment we discovered, which we think plays a role in amino acid homeostasis. But really the big protection mechanism and regulatory mechanism for cells, I'll come to at the end, is the MVV escort pathway, which if you feed them high amino acids, they will ubiquitinate and downregulate these transporters. And so cells are actually smart enough not to kill themselves. And what's really cool about that, there's really cool crosstalk I'm going to come to. So if you impair vacuole function, that sends a signal to then downregulate these transporters of the plasma membrane. And so if we feed them like a lot of lysine, for instance, no, it doesn't kill them. But the reason is because these transporters get downregulated. So there's lots of work in our lab. A postdoc, Kylie Jacobs now, is using mutants that have they're defective in their ability to regulate plasma membrane transporters. So they're always stuck on the surface. And those mutants are extraordinarily sensitive to amino acids at micromolar levels. It will kill them. So yeah. So the idea is that cells, they have a lot of defense mechanisms that we have to sort of peel away to understand the phenotypes of intracellular amino acid toxicity. Great question.
We've got a couple of questions in the chat.
Awesome.
Good.
I'll take a look. So from Jason Shapiro, have you looked at the levels of these amino acids in yeast that are not deficient for biosynthesis?
So the reason I asked this question is I work a lot on, so I'm from Hossein Al-Rahali's lab. I think we've met once. I know a few other people here. I work on mTOR signaling and iron deficiency. And one of the things that I found was that amino acid transport is actually regulated by iron levels. And that's one of the mechanisms it works on to regulate mTOR, it's specifically through leucine. But a lot of these yeast, it's interesting that, like, for example, the leucine biosynthetic enzyme in yeast is an iron-dependent enzyme. And so I found that there's this major connection between leucine and iron, but I was curious, most of these yeast are deficient for Lu, Met, His, all those kind of things. Is this really a transport issue? Is this a biosynthesis issue with iron deficiency in conchycin A? Like, have you looked into that?
Yeah, great question. So we do almost all of our work in prototroph yeast strains that don't have these auxotrophic defects to get around these kinds of complications. Because yeah, as you said, it's like Lu1, right, requires iron. So it's highly complicated and interconnected. And so we've ruled out some of those things by just using prototroph strains. Yeah, yep. Great question. And also in the chat from Caroline, are levels of polyamines like spermidine rising? I don't think we've looked at that, but we should. It's a really that would be really cool. We have.
I mean, that's sort of a long-standing concept of stress response, is that you take your amino acids and you pull the nitrogen out, or you use the glutamate and glutamine for all of the nitrogen requiring biosynthetic processes in a yeast. So those are sort of like all-purpose amino acids that are also used for storage.
Yeah, and I think that the thought would be that it probably is, because whatever, you know, when we look and see, you know, like, and we've done metabolic tracing here too with labeled glucose, and there is a huge amount of flux under these conditions of of the amino groups, you know, being transferred from amino acids onto glutamine, glutamate, but then downstream of that, we haven't followed where that goes. And I think you're probably right that there's going to be a lot of downstream metabolites accumulating there for stress responses here that help them cope.
Thank you.
Yeah, sure. All right, I want to make sure, because I'm doing this in more of a discussionary type format, I want to make sure that I get to some of our newer stuff today. Let me see where I want to get to here. Maybe I'm just going to skip into this model because I think it won't give us enough time to really get into this. So coming back to this concept, I'll just finish this. So we have this idea that amino acids are problematic for the cell. And what we actually began to wonder from this is, are these amino acids actually driving the mitochondrial deficiencies that are occurring? This isn't exactly the way this happened. This actually came through genetic screens. And what we discovered is that if we overexpressed amino acid transporters in aging cells, where there was still a proton gradient, but it was just low, this was actually sufficient to prevent mitochondrial dysfunction, suggesting that, you know, perhaps if we could reorder or rearrange where amino acids were. The tricky thing is that we haven't been able to effectively measure this. Obviously, work from David Sabatini's lab has pioneered, you know, isolating organelles and looking at the way metabolites are organized at a organelle level. We Osiris Martinez Guzman, who was a postdoc in our lab, she just moved on to a position at a company, was actually working on this in yeast and trying to come up with really nice ways that we could pull out different organelles and analyze metabolites, but we aren't quite there yet with it. But the cool thing about all this is that this is showing you here that, again, this is just published data, that this phenotype of VATPase deficiency driving mito problems. So again, you can't grow on glycerol here if you lose VATPase function. It turns out this only occurs in rich medium where there's high amino acids present. So coming back to the question from Jason, when we work in prototroph strains, if we go to a minimal media with no amino acids, or even just a low amino acid medium, it turns out that it totally uncouples this relationship. So you can impair vacuole function and lose acidity, and mitos are just fine. They keep on churning, they keep on plugging, respiration is not inhibited. What's not shown here is then we can add different amino acids back in, and we actually know that the most problematic amino acid for mitos is cysteine. And what we went on to publish in looking at this, and the question became, okay, how does this actually happen? What is it that amino acids and cysteine are doing? And this was the focus of our work, and what I won't get into all the mechanisms of today is that cysteine, we think, ultimately ends up impairing intracellular iron bioavailability. And so this is something that I think has become really interesting for us and pulled us totally into the realm of metals. And you know, I was not in this field at all before. Luckily, being at Utah, I've had so many great colleagues to discuss and work on this with. And so we collaborated with Dennis Wingy on this quite a bit, investigating this. And so I'm actually not going to talk a ton about this today, but I'll summarize this in just a couple of slides so we can stay on time with focus. And so what becomes clear is that if cysteine is not properly controlled within cells, we think it builds up, the working hypothesis, it builds up and then it somehow interferes with iron homeostasis. We've worked a lot with Lori Jackson and the Iron and Heme Core to measure iron in these cells, to look at a lot of different phenotypes in terms of what's going on. And all I can really tell you is that iron levels are actually normal, if not elevated under these conditions in VATPase mutants. And this has been demonstrated by others as well, Patty Kane's lab and a few others. However, one of the things that they exhibit is constitutive iron starvation that turns out to be amino acid or cysteine dependent. So even though iron levels are normal, I'm not going to show you this here. I'm going to skip over this here. Let me skip around just a little bit. I'm going to keep this going here. So this was nice work several years ago from Patty Kane's lab, where they had actually shown that loss of acylar ATPase activity in yeast leads to a constitutive iron deprivation phenotype. So what we came at it from was that there's a major amino acid problem, and Patty's lab had shown that there's a major iron limitation problem. And we sort of just connected the dots between these two and investigated this a little more thoroughly. And basically what becomes clear is that if you measure, again, iron, there's normal iron levels in the cell, but that iron is not bioavailable because the cell is certainly perceiving, and there's actually a functional decline in the levels of iron sulfur cluster proteins and the ability to make respiratory chains. That in turn turns on the well-known in yeast, the AFT transcription factor regulon, which you can see nicely here by qPCR in a wild type cell. If you treat them with concatamycin A, you get very robust induction of the iron regulon in terms of increase in transcripts and genes that are important for iron acquisition. This is totally dependent on this transcription factor AFT1. We can do this other ways too. We've also built, this is again working off work that Patty Kane's lab had done, taking the promoter of one of these genes, FIT2, and hooking it up to GFP to make a reporter that we can use by flow cytometry or Western blot analysis. And we can see that VATPase inhibition leads to really nice and robust activation of the iron regulon, very similar to iron chelation. And this is all AFT1 dependent. And it also turns out, and Patty showed this as well as us, that AFT1 is essential in VATPase deficient cells for survival. And so really what it came down to in digging into the literature is that we saw two different phenotypes going on. VATPase mutants had extensive changes in amino acid metabolism and miscompartmentalized amino acids. And at the same time, they also showed huge changes in iron bioavailability, even though iron is actually present. So somehow the iron isn't particularly usable in these cells. And really what we're seeing through screens is the predominant phenotype of a VATP deficient cell is iron starvation. And I'll get into this in a second, but this is also very true in the mammalian cells and a very important role for lysosome acidification. I'm going to skip back a slide here to show this because I think this is pretty cool. I did this probably a little bit out of order, but you can see this here. So if you add iron back to VATPase mutants, it very nicely rescues their growth, much like lowering amino acids, as I showed earlier. So we have two different ways to do this. We can lower amino acids, or we can give them more iron. And that's going to be the theme throughout the talk today. And I'll come back to this in a second. You can also do this really nicely with this visual assay. So I showed you these movies earlier. It's the same two movies, where if you treat cells with VATPase inhibitors here, that the mitochondria become fragmented. However, if you supplement them with, in this case, it's 1 to 2 millimolar ferrous ammonium sulfate in the media during this time point, we know that the vacuole is still deacidifying here. But what's cool is that, in showing with the rescue of growth on glycerol, is you also see a reasonably strong maintenance of mitochondrial structure. And there's also maintenance of mitochondrial function. And not showing here is we can supplement with iron and maintain mitochondrial morphology throughout the aging process. So sort of linking back to those phenotypes. So really in coming down to this, we got to this point. And so this is an interesting question, I think, for us. So we had an amino acid issue and we had an iron issue. And the way this is drawn here, and we went through this in quite a few steps, and I'm going to just show this in just one piece of example summary. It turns out that the amino acid issue, and specifically cysteine, we think is actually upstream of the iron. So it actually causes the iron lack of bioavailability. And just to show you one example of that, you can see that here. So if we look at our iron reporter, FIT2 driving the expression of GFP, so this is an indication of whether cells are iron starved. We treat cells with VATPase inhibitors in the presence of high amino acids, and we get really nice induction of this reporter. So the cells are highly iron starved. It causes all of our phenotypes. However, again, these are prototrophic strains, so we can do this in a minimal medium or a media lacking amino acids. So we're not giving them any extra. And basically, there's no mitophenotypes and there's no iron starvation. And we've done this several other ways as well. And then what we can do is go back in and add in individual amino acids or amino acids in the form of peptone. But basically, if you add them all in one at a time, what ends up being the problem is cysteine. And so our working model, as I mentioned right now, is that one of the major roles in the vacuole and lysosome in cells is to, especially in yeast, is to compartmentalize amino acids. So they come in, they need to be organized. And if you see disruptions in this, could be due to aging, could be due to mutations, or in our case, we oftentimes, again, use chemical inhibitors. This leads to alterations in amino acid homeostasis and such that we think that cells are dealing with at least transient changes in amino acid levels. And we think one of them that's particularly bad is cysteine. And ultimately what cysteine ends up doing to harm cells, and we have other pieces of data I don't have time to show you today, that we think cysteine somehow interferes with the bioavailability of iron. And so what I'm not getting into, and what has been a big question for us for a while, is how does cysteine do this? And cysteine obviously is very good at chelating metals. There's a high likelihood that part of what's going on here is there's cysteine mediated iron chelation, this sort of improper chelation that's happening. We weren't really able to completely demonstrate that, and it's still very much an open question of what cysteine is doing here. What we did have quite a bit of evidence on is that there is some link here to oxidative stress that we also don't quite understand, and by which cysteine is interfering with iron homeostasis. And of course, obviously, cysteine is a major component. Its movement into mitochondria is a major component of iron sulfur clusters themselves. So there could be something interesting there going on with, even NFS1 and having too much cysteine flowing into iron sulfur cluster biosynthesis. So I think that's a very interesting area of investigation that honestly our lab right now, I think we've moved in other directions and we're really not focused on it. So if anyone is ever interested in sort of looking more closely at detailed mechanisms of how amino acids like cysteine in a cell could interfere with iron, we'd be always happy to collaborate. And one question that comes up a lot is what about other metals? And so cysteine has affinity for other metals as well. And we do see effects. So we focus a lot on iron, but there is also effects going on, I think, with copper and zinc and other metals. And we also see that other amino acids, we think, and this has been reported before, and we see the same thing like histidine, we think actually causes a problem with things like copper as well. So you can have similar types, and histidine is very highly stored in vacuoles. So similar types of things going on with other amino acids as well.
Can I ask a quick question?
Yeah, totally.
Sorry, I know you're trying to move through this, but do yeast,
we're good on time. I just want to get through that first part. So,
do yeast have homologs to the steep proteins in mammalian cells? And if so, do steep proteins require cysteine and all? Because the iron sulfur cluster, if cysteine was limiting, your iron add back probably wouldn't work as much. So and I do believe in maybe the chelation thing, but I'm wondering, so in mammalian cells, the steep proteins are necessary to reduce the Fe3 to Fe2 to export it from the lysosome. And if you knock out steep proteins, basically you get this lysosomal iron accumulation and cytosolic iron deficiency. The whole thing shuts down. It's very similar to your process. So I'm wondering if there's this unknown role for cysteine in regulating homologs of steep, if yeast have it. I don't know if yeast have them.
It's a great question. So Dennis is shaking his head. I actually don't know. This is a little bit out of my wheelhouse. Do they have, the people who are more aficionados, do yeast have them? Dennis, you're muted. Oh, he's trying to answer. Dennis, you're muted. We can't hear you.
They do have lysosomal vacuolar reductases.
Yeah, there's 7 different steep proteins in yeast.
Awesome. And so I think this is one of the interesting things for us. Obviously, there's a multitude of ways from work from Jerry's lab as well that and I'm not getting into all the details of everything we've looked at here. Obviously, the VATPase is important for FAT3 dependent iron transport. So there are multiple more simple explanations too. And so what's interesting about this is I think, and this is just going to highlight some of links to mammalian cells. I think the role of amino acids and cysteine in this has been very interesting for us, and it's kind of an extra layer. But in terms of losing VATPase function, it's going to hit iron homeostasis at multiple different points. And this was, we coordinated quite a bit, both with Tiny, with Nuno Raimundo's lab, as well as Kervanch Borsoi's lab, and sort of discussing all these findings. And we actually published Kervanch and I co-published this together, where the really cool thing coming at it from a totally different direction from what they had seen is that, yeah, VATP, the essential function of acidification of the lysosome, right, is really maintaining iron homeostasis. So one of the things to keep in mind then is what happens downstream of this. And the more we've begun to look at this, as I mentioned, that every iron-dependent process that you could imagine is downregulated in a VATPase deficient cell. Ergosterol synthesis, unsaturated fatty acid biosynthesis, that we think there's issues with translation and DNA repair. And so really losing this, has a major effect, we think, in both yeast and mammalian cells on iron homeostasis. And I think if we could get one thing across, it's that, thinking about lysosomal dysfunction in the context of disease and aging, it's complex, right? You have effects on signaling, you have effects on nutrient homeostasis, amino acids, you have major iron deficiencies, and all these are going to be contributing to the different phenotypes that we see in cells. Caroline had a question in the chat. Have we looked at glutathione? Yes, we have, and we've done a lot. This has been a couple years now. I'm trying to remember, like pre-COVID, so my COVID memory is erasing it, but we explored it quite a bit and didn't see, like, we can add cysteine to cell. Just adding cysteine to cells causes a lot of these iron issues, but adding glutathione doesn't. And we looked a lot at the concentrations of it. It was actually increasing, I believe, under VATPase impairment. And what we tend to see is what cells like to do is they move these amino acids into pathways, as you said, like polyamines or something like that. And we think that a lot of cysteine is actually moving into glutathione as a way for the cell to actually potentially rid themselves and minimize cysteine concentrations.
I was mostly thinking in terms of iron sulfur cluster assembly, if that's the major phenotype associated with your mitochondrial dysfunction, because you have to use glutathione to build iron sulfur clusters. It's kind of interesting. Karen Outen presented some work a few months ago where they were looking at ways that yeast can survive without building iron sulfur clusters. One is with high levels of cysteine. Now, when I say survive, not building iron sulfur clusters, survive a specific defect in the iron sulfur cluster assembly system in the mitochondria. And it wasn't cysteine per se, but it was something that was derived from cysteine. We were kind of wondering whether it's actually homocysteine. So it seems like glutathione synthesis and glutathione levels are sort of intimately linked with the process of building iron sulfur clusters and chaperoning, I hate to use that word here, but iron around in the cell.
Yeah, no, it's a really, is it part of, is it a ligand for the bioavailable iron pools? And Karen and I have talked actually quite a bit about her data too.
Yeah.
And we've talked about it extensively, and it's in knowing that there's some sort of interconnectedness between this, but we haven't really been able to nail it down and what is it. So I wouldn't be surprised if, yes, it's some normal, important functional role for cysteine in this, but then it becomes kind of out of whack when you don't have these, the ability of the vacuole to serve as a buffer for it. You can imagine it's altering amino acids and iron homeostasis simultaneously, right? So we've looked, it's somewhat puzzling because we can add N-acetylcysteine and it actually suppresses these phenotypes, concatamycin A-induced iron starvation. And so that was something I think we had in our original paper, and reviewers asked us, they're like, we don't understand this at all. Why don't you just take it out? So, there's certainly a very deep, intricate puzzle here that we don't fully understand the mechanism of it.
So Adam, does the low iron availability affect enzymes like ribonucleotide reductase?
Yeah, we haven't measured it. It affects, certainly we looked at aconitase activity and others have as well.
But enzymes that don't require iron sulfur.
Yeah, it does. Yeah, so it's not just an iron sulfur cluster defect. Yeah, so TK in the lab has done a lot of lipid profiling. He's looked at this quite a bit. And yeah, any iron or heme or iron sulfur cluster. So di-iron is down, heme is down, and iron sulfur cluster. So there's a general lack of iron bioavailability for any iron dependent process. Yeah, so it's also not specific to iron sulfur cluster. Great question. And there's a question in the chat too from Joseph. Has this been demonstrated in any mammalian cell lines? So what has been, so this is a really great point. It's something that we would love to do. So what's cool in mammalian cells is it's been shown that very well that VATPase inhibition, and we've done some of this work in our cell lines, that it leads to iron, similar phenotype. You get cells have iron around, but it's not bioavailable. And it leads to what I didn't show today is an inability to build respiratory chain complexes. So you can't, and again, a number of other iron dependent processes are down. What hasn't been linked at all is whether or not there's a role for any sort of role for amino acids in this. Obviously in the mammalian field, then it gets very interesting and complicated with things like ferroptosis and connections between iron and cysteine, and the transporters are all different. But the one thing that we're really fascinated by, so this came out of work again from David Sabatini's lab, when they initially pulled on lysosomes to do metabolite profiling. The yeast vacuole is well known to have a lot of amino acids typically. The mammalian lysosome seems to, at least from initial studies, operate in a bit of a different fashion, where it is wired more to spit those amino acids out and not serve as a reservoir. Unless you do things like inhibit TOR or inhibit the VATPase. It's actually somewhat wired backwards. Then, so those are actually required for amino acid export. But the one amino acid in the mammalian profiling that was, and I've talked to Munther, who has a lab at Stanford, who's done a lot of this work. One amino acid that is VATPase dependent concentrated in mammalian lysosomes is cysteine or cysteine. And recently also an additional amount of work identified the transporter that's important for this. So that has now unveiled. We haven't done this yet, and we would like to. It'd be cool, but we just haven't had the manpower, is to go in and see if, altering that transporter, the mammalian cysteine importer on lysosomes, has any effect on iron homeostasis and what it does. I would bet that it would, and it would be a really interesting avenue to go in. And again, something we're not doing, and would be happy to explore with somebody if anyone's interested. Great questions. So what I'm going to do now is just talk about, so I'm going to spend the last, and since we've been doing questions throughout, I'll just sort of go up another 10 minutes or so, and then leave a little bit of room for questions at the end. But I want to talk about some of the newer things we've done. So this first part of this is really talking about, I know for a lot of you that was new, so getting into some of the things we've done from this. So in identifying this really interesting link between these organelles, again, what I've talked about, what we've become very interested in is understanding now the role of the vacuole in what it plays in general in the spatial compartmentation of amino acids, and how loss of these pathways ultimately impacts the cell. But one of the things that we did was sort of use a simple genetic screen. And we said, okay, if cells lose VATPase impairment, there's amino acid issues, there's iron issues, mitochondrial deficits, but the cells are surviving on glucose. I want to be clear. If we don't require mitochondrial respiration, cells can adapt. And so we just became very interested in trying to understand what are the ways in which cells have to adapt to this. And the idea is that during times like aging or VATPase dysfunction that's going on, perhaps in different diseases, maybe we can identify pathways that are critical to helping the cell maintain amino acid and iron homeostasis. And we thought that it would both pull out pathways and also help us understand a little bit more of what's going on. And so what I want to do in the last 10 minutes here is just tell you some of our interesting results from some genetic screens that have been carried out by several people in the lab, including TK Cody, and some of the follow-up work that he has done along with another graduate student, Kevin Choi, in looking at pathways that become essential in the absence of a functioning vacuolar ATPase. And what's kind of cool is this is what Kavranch Boursoy's lab really did in mammalian cells. They use CRISPR screens to do this, to look for things that, same thing, the idea is synthetic lethality between loss of the ATPase function. But it was a very targeted approach because there the libraries are focused on metabolic genes. But in this case, what we ended up doing was we took the yeast as a starting point. We've done this several different ways now, is we took the yeast non-essential deletion collection and we did a very simple, very qualitative, kind of old school screen here that was really easy. And we just took this collection and we plated it. So 96 well plates. You actually have to grow them in liquid exponentially a little bit first to really see nice, strong phenotypes. And then we plated them either on YPAD, just normal rich medium, or YPAD plus concatamycin A. And so these strains, because this is a deletion collection, are actually oxytropic strains. So they do have some of these amino acid deficiencies. So just keep that in mind. And then what we did is just look for interesting comparisons. So we went through this and scored this by 2 independent people, coming up with just categories of 1, 2, 3, or 4. And we've done this screen several times on both the haploid and diploid deletion collections. And what you can see here is just an example of a phenotype. Is here's a number like this one right here, right? You can see it grows nicely on YPAD, but is highly dead in the presence of the ATPase inhibitors. And so the idea was just to find genes that become conditionally essential in the absence of vacuolar ATPase function. And probably is not wildly unexpected. We got a lot of genes. So we got like 600 genes that become at varying degrees of essential. The vacuole does a lot of things in cells. And so what are some of those things? And this is what's really cool. And this is where we're moving in the future. So some of them made a lot of sense to us right away, and they're really nice controls. So this is just spot assays of things that have been remade and re-deleted in our lab and confirming them. So as I showed you before, so here's AFT1, the iron-dependent transcription factor, which becomes essential, is necessary to survive loss of the ATPase function, both with concatamycin A and also if we get rid of it genetically. We had shown this in our other paper before. FET4, as well as some oxidative stress response genes, are also required and important to survive the ATPase dysfunction. But there's some other interesting groups that came out of this, and I'm also happy to share this list where TK is putting together a manuscript on this right now, and we hope this will be published as part of that, because we just don't have any capacity to work on a lot of these things. But a lot of what we see is there's a lot of chromatin remodeling and DNA damage repair type enzymes that become more essential in the absence of a VATPase. One thing that we haven't worked on, but what's really interesting, there's a complex called the Hermes complex, which is involved in ER mitochondrial tethering, which also becomes highly important in general in the absence of a VATPase. And also a number of cellular proteases. And I'm going to talk about this. I may show a couple pieces of data on this just from the end. TK has actually been highly focused on, this is the mitochondrial lawn protease, PIM1. Which we see becomes essential in the absence of a functioning vacuolar ATPase, and we think plays an important role in actually maintaining iron homeostasis. What I'm not showing you here is what's pretty cool is almost all of these synthetic lethalities are rescued by either lowering amino acids or by giving iron back. So we think that predominantly of those 600 genes, we haven't tested all of them, but a large number of them are connected to this amino acid iron dyshomeostasis issue. Some of them making a lot of sense here, like an AFT1, some maybe not as clear or evident in terms of chromatin remodeling or DNA repair, but that could also be linked to iron-dependent enzymes involved in those processes as well. So I'm just going to spend just a couple minutes here talking about finishing with just one interesting thing. I don't think I'll have time to get into TK's work on PIM1 LOM protease today, but this is work that TK had started, and now Kevin has taken over and is working on it. But one of the groups that I didn't show you that we became very fascinated by, and this comes back to thinking kind of about bigger picture here, is what we notice is that a number of genes shown over here on the left become highly essential in the absence of a functioning VATPase. These are actually all proteins that are involved in the well-characterized multivesicular body escort pathway. So this pathway is very well characterized, and all of these different genes play roles at different steps here, but ultimately what they do is they're involved in, under elevated amino acid conditions, providing feedback and ubiquitinating amino acid transporters and removing them from the plasma membrane for degradation. So this kind of comes back to, I got into this, I jumped the gun a little bit from a question and got into this a little bit earlier, but this is going to show you some of the data here. So we saw that these genes become conditionally essential. So that suggested that perhaps this pathway is actually highly active in the absence of VATPase function. This is sort of an interesting idea because to degrade these transporters, you would think that you would actually need vacuole acidification. So why would you actually activate this? In yeast, this is where it's a little bit different than mammalian cells. So if you deacidify, these things are actually pretty well efficiently still degraded in the vacuole. So you don't completely need acidification to have some degree of protein degradation in vacuoles. But what's really cool here, and this is just one small example of this, we've done this extensively. So there's a number of different amino acid and transporters across the plasma membrane. Patty Kane's lab showed this for PMA1, the proton-dependent ATPase, and what we ended up looking at was a number of different transporters. And so what you can see is that normally in an untreated yeast cell, they're expressed highly at the surface, and there's some of it that diffusely localizes to the vacuole. This is very nutrient dependent. So these cells are grown in rich nutrient media. And what it is there's a balance between, there's a lot of this getting to the membrane, but there is some constitutive turnover that goes on. However, when you add VATPase inhibitors, what happens is the amount, and I'm not showing the quantification here, but we've done that as well. The amount on the plasma membrane becomes reduced, and a large amount of this transporter, this is GFP tagged, it ultimately gets delivered to the lumen of the vacuole. The transporter gets degraded and free GFP accumulates here. So this is a very well described assay that's used to show induction of this system and turnover of plasma membrane nutrient transporters. Just to give you an idea, so if you knock out one of the genes required for this pathway, if we treat cells with VATPase inhibitors, this transporter stays locked at the surface. So there's a lot of data I'm not showing you here, but I just want to kind of give you a working model of what we think might be going on, and then I want to finish up and take any other questions and think about where this is going. So this is kind of getting into the bigger system here. So we talked about the VATPase playing a very important role in regulating amino acid pools. Well, it turns out, and the idea is that if this is impaired, this starts to lead to amino acid problems and iron limitation. But the cells adapt. They turn on the iron regulon. They do other things. But one of the things that we think they do is they then ubiquitinate these transporters and turn them over. And if you don't do this, it now leads to synthetic lethality. So the idea is you need both of these systems. And so we're going through now and testing this hypothesis. And one of the ideas is that if this is leading to synthetic lethality due to too many amino acids, that we should be able to suppress this by removing amino acids from this condition. And that's absolutely the case. So you can do this in low amino acid condition with prototrope strains. And again, much like the mitophenotypes, if you remove amino acids, there's no growth defects at all now in the combined absence of these mutants in addition to VATPase inhibition. So the idea is that the amino acids are really a problem here, and they're accumulating. The cell has multiple ways to try to handle this. And what's really cool is if you go one step further, this is again linked to iron homeostasis. So here's a similar swath of mutants that are sensitive to VATPase inhibition, and you can lower amino acids. What you can also do is supplement with iron. And so what we think this leads to is kind of this interesting model where if you start to put it all together, amino acids come into cells, right? And their homeostasis has to be maintained. And there's a balance in this. They're utilized for metabolism. They're utilized for protein translation, and a large amount of them are stored. And what happens is if you start to lose these systems, I didn't show you this today, but if you start to impair protein translation, this is a really well-known way to boost amino acid pools. This also leads to iron homeostasis issues. So we think there's this really intricate connection between amino acid pools, particularly cysteine. There may be other ones involved too, and metals, and that cells undergo a variety of mechanisms to help control amino acid homeostasis. And if you begin to lose these, what the ultimate impact begins is you have amino acid issues, but that really leads to metal deficits, iron as well as others. I should say that there is some role for threonine and also serine in impacting iron homeostasis that we haven't yet explored. But where our lab is now moving in the future, I'm going to skip a few of the last data slides here just to finish up with acknowledgements. It is really what we do. We have two sides that are kind of, as I mentioned before, we'd be interested in, I think, collaborating and continuing on the cysteine iron specific connections and how all that's happening, and is there any relevance to this in human disease. The other of what we're doing is thinking about the 19 other amino acids. And even though this is a metals talk, it turns out that a lot of the phenotypes that we're seeing are not metal related. And so some of the amino acids have, we think, interesting roles in other places in the cell, like impairing protein translation, or perhaps other effects as well. And so if you kind of look at a sort of global idea of what we think happens as amino acids come in, there's this interesting balance, but if this is out of whack, this leads to many effects in the cells, a lot of them iron dependent. So we think DNA damage might result, which could be resulting from iron limitation, mitochondrial dysfunction. We do see effects on translation. We're starting to investigate that now. We don't yet know whether this is connected to some of the known roles for iron in translational regulation, but that's something we're interested in investigating. So this is just summarizing that here. Amino acids are compartmentalized. Loss of vacuole function alters them. Elevated amino acids impair many iron-dependent functions. And the escort pathway, and what I didn't talk about today, is that there's another protease that TK has been working on, PEM1, which he's told you all, a lot of you about before, we think are important in maintaining iron homeostasis in VATPase deficient cells. So with that, I just want to, I'll take any further questions if there's any. I'm happy to stick around and discuss too. Obviously, I want to thank the lab here. This is an older picture, pre-COVID. We've taken a new one, but I didn't adjust it yet. The work I talked about today was by Casey Hughes, TK Cody, and Kevin Choi, and some of our wonderful collaborators here, especially a shout out to all the folks in the CIHD community, which has really adopted me over the years, even though I'm not a complete metal biologist. We have so many great interactions with Dennis and Diane and Jerry and John and others, and also really great collaborations with the core facilities, James Cox and Lori Jackson, especially. And I also want to thank our funding sources, including pilot grants from the CIHD. So thanks, and I'll take any further questions if there are. I know I've answered a lot throughout.
So Ola's got a question in the chat.
Awesome. Hi, Ole, good to see you. So reading part one of your talk, if you looked at these effects in the context of mitovacuole contact sites, were there any changes in number of intensity of V clamps in conate-treated cells or any VATPase mutants? Yeah, really great question. So there's obviously physical connections between lysosomes in mitochondria and vacuoles in mitochondria. So V clamp, for anyone that doesn't know, is a protein complex that physically connects vacuoles to mitochondria. We have looked at V clamp to some extent. And I want to make sure I get this right. We didn't see any obvious effects on this pathway, I think. It's not something we're actively working on because I think it was kind of one of those one-off things that we thought the exact same question. We looked at it, knocked out V clamp components, but there's not an effect on iron metabolism, I don't think, not anything that we followed up on, but there was some effect of V clamp in terms of VATPase deficient phenotype, where the tricky thing is, I think what we did at the time was use two different mutants. There's some of the V clamp mutants are also involved in the hops vacuolar fusion complex. So we mutated two different components of the hops complex, one involved in V clamp and one that wasn't. And I think we saw differential sensitivities there to some of these issues. And we probably did this 6 or 7 years ago, but there wasn't anything major that I looked at. And V clamp sites, we haven't completely looked at to see whether they're changing or adjusting, but I wouldn't be shocked. There's got to be some really cool communication here that's going on at these local sites. And it's something that we've talked about looking at, but just haven't. Yeah, really cool question.
Yeah. So did iron rescue any of the V clamp phenotypes, rather than thinking that the V clamp was creating the iron problem? Was the iron problem creating a lipid problem? Is that a question that makes sense?
Yeah, is the iron creating? Number one, the iron problem is certainly creating a lipid problem. We have worked again with James in the core, and so there is in VATPase mutants, there is a large deficit in unsaturated fatty acid biosynthesis through the OLE1.
OLE1, yeah, okay.
That's impaired, and so you have increased linosterol. Anything that's hemed, all the P450s are inhibited. So there's alterations in sterol synthesis. So yeah, so a lot of the phenotypes, some of these things I think are probably highly interconnected, and that could easily play a role too. Like you get amino acids that affect iron, that affects lipid, that then in turn affects mitos. There's lots of things going on here, I think, that becomes complicated to dissect.
Yeah. Oh, and I was just going to share with you. So we've been working in mammalian cells, obviously looking at intracellular iron trafficking, and we see a whole slew of chromatin remodeling and DNA damage phenotypes.
Do you know much about? So our assumption has been, I don't know that we haven't. I guess we've seen it over and over again. They become essential in VATPase mutants. And so we're assuming that there's some sort of chromatin remodeling adaptation going on, whether that, we haven't looked. Our thought was maybe it's for efficient expression of the iron regulon in yeast at least, but we haven't paid attention to that or looked at that yet. But
what we see is more direct. I mean, you can iron starve a mammalian cell and then create a lot of DNA damage and change the stability of a number of iron-dependent chromatin remodeling and DNA repair proteins.
do you think some of it is there? Our assumption has been that there's probably a fairly, and TK's looked at this a little bit, there's sensitivity to ribonucleotide reductase inhibitors. Those steps are certainly impaired, we think, to some extent because of the low iron.
So some of it seems to be iron sulfur cluster assembly, and some of it seems to be metallation of mono and diiron sites in histone demethylases.
Oh, cool. Well, we're happy to share. We haven't, if you're ever interested in the data or seen it, we haven't, again, something we haven't.
I'm just sharing is like we bumped into the same phenotype that, seems to be worth examining. Yeah.
Any other questions?
Yeah, I have one. This goes back to the very early part of your talk, and I've never thought about this before. You showed in old cells that the ATPase gets impaired. And the curious thing is that you can suppress it by overexpression. And I think I remember from previous data of yours that it's different subunits you can suppress it. So the question is, how do you think about that? Because the way I think about that suppression is there must be some DEG going on, maybe from the cytoplasmic proteases. Yeah. And so is that having a role on mitochondria? If the cytoplasmic proteases are getting activated in age cells, do they have a deleterious effect on mitochondria that also contributes to the iron impairment?
I think I understand, at least I understand the first part of your question, that, yeah, like there's this, when we did suppressor screens, there's not much that works. So in terms of VATPase, you can overexpress the catalytic A subunit, VMA1, and that works really well. But many other ones don't. And actually some are inhibitory and problematic. The other thing that works well that I didn't show, the one other one we get was VPH2, which is a chaperone involved in assembling more. That mutant, and when we looked at VATPase levels, there was just more VATPase in the VPH2 overexpression. The VMA1, no. I spent a long time actually evaluating, like, is there something wrong with VATPase? Is it not assembled? And it all looked normal. And so there was never anything that we could tell was actually wrong, leading to deacidification. Part of the issue actually comes, there's another study from Dan's lab after I left that showed that PMA1, the plasma membrane ATPase, actually accumulates on aging mother cells as they age. And so it actually pumps protons out of the cell, and there's a bit of a proton issue that can lead to diminished vacuolar ATPase. So the overexpression of VMA1, one idea that we had a long time ago, there's a cysteine, this could come back to redox, there's actually a cysteine on that protein that's conserved across evolution. It becomes highly redox, it's redox active, and it will become oxidized, and that will impair its activity. And so we were wondering if something like that is actually going on with aging. And we never actually tested that. You can make a point mutant in that cysteine and it's totally normally active. We always wanted to do that and see if you could maintain vacuolar acidity, but we never did it. The degradation part of it, I don't know. Yeah, there's certainly a lot of things, like when mitos are impaired, a lot of these mitoproteases that I didn't get into today, we think some of them are actually accumulating outside of the mitochondria. So things become a bit haywire and a bit of a mess. And I think there's a lot going on here that we don't understand.
So Adam, this is Ole again.
Hey, Ole.
Question. Hey, good to see you. So I'm trying to think. So this weird phenotype we've been sort of chasing a little bit for, some of the Rho zero mutants or Rho minus mutants being able to rescued by RNR1, so ribonucleotide reductase or expression. I wonder if that could be the piece of the same puzzle.
You said you can rescue them with RNR overexpression, is that
right?
Yeah,
you can stabilize those, yep. And another thing that rescues them is a glutamate transporter. It's this YMC2.
Yeah, I mean, certainly, like, because in those petite mutants, right, there's so many rearrangements in both iron metabolism. There's so many deficiencies in that, and also amino acid metabolism, right? There's also an interesting connection too. I don't know that in that case, I think, I'm trying to think, we can't rescue the ATPase deficiency with RNR overexpression. TK did this. It actually makes it worse, almost as if we were like, maybe it's soaking up the iron, it's making it even less. So we tried a few of those things, but I do wonder too, what was I going to say? I had a train of thought that I am losing. What was it? I'll think of it in a second with the ribonucleotide. It's this interesting finding too that petite mutants are actually rescues the VATPase. Corey Dunn showed this a long time ago. So the if you're mitochondrial petite, then treating with a VATPase inhibitor like Con A is actually beneficial. So there's some total change in the wiring here that's really interesting. And we've always suggested maybe it's some of these severe metabolic and chromatin adaptations that are going on in petite mutants. It's changing the way the cell survives, but would be really cool to look at. So I don't know. I don't have a good answer to your question, but
that's fine. Thank you.
So in your conditional essential set of genes, you got SOD1, and I'm wondering if that implies oxidative stress. Do you know whether you can suppress that with DTT?
I don't think we can. I think we've tried DTT. We tried a lot of antioxidants. The only thing that we can, you mean the SOD1 phenotype specifically?
Yes.
That we haven't looked at. We haven't looked at what we can suppress it with. But there is definitely oxidative stress going on, and Patty's lab had shown that as well. And what we did show is that the iron phenotypes, so the cysteine-mediated iron inhibition is oxygen dependent. So if we do that anaerobically, you do not impair iron homeostasis anymore. So there's some really interesting connection to oxidants in this that we do not we've never been able to completely understand it. Yeah, but it's fascinating. So obviously mitos aren't working because you're anaerobic, but the iron AFT1 regulon is totally shut off. They're not iron starving anymore. And you can add BPS under low oxygen and return it back on. So it still works, but yeah, you can suppress VATPase iron phenotypes with anaerobic conditions. Well, I should probably let folks go. I'm happy to stick around a couple minutes if anybody else wants to, but that was awesome. It was a really fun discussion. So thanks for interrupting me a lot today, and it was great.
Great talk, Adam.
Thanks, Adam.
Good to see everyone.
Nice to see everybody.
Really enjoyed it. Thank you.
Thanks, Caroline.
Take care. See you, Jer.
See you, kid. Stay well.
I'll do it.
Thanks, Diane.
Yeah, thanks, James, very much. And thanks,
Yeah.
Adam. I've got a lot to think about.
I'm happy that Diane, we should dive in more. We got a lot more, obviously, that I didn't get into today, but
Yeah, there is a lot to talk about. Okay, I'll quit the recording.
See ya.
See y'all. Thanks
Thanks.
again, Adam. That was great.
Thanks, Adam.